Hi, everyone, and welcome to DNA LC Live. My name is Megan. I'm an instructor at the DNA Learning Center over at Cold Spring Harbor Laboratory. Thank you so much for joining me. What we are going to discuss today is the diversity of life. We are going to take a look at plant and animal cells under a microscope, and we're also going to have a quick discussion on taxonomy and classification. So, there are so many different organisms on our planet, and in order to better understand them, it's very important that we are able to organize and classify those organisms. So there was a system developed in the 1700s by a scientist named Carl Linnaeus, and he is known as the father of taxonomy. Before I jump into explaining and showing you his system of classifying living things, we're going to do a little exercise that's going to relate this idea to something that's probably more relatable. So I want you to think for a moment about your favorite store. Okay, think about what your favorite store would be. You could even close your eyes and pretend you're walking into that store, which we most likely can't do at this point. So think about that favorite store. All right, maybe your favorite store is a clothing store. Maybe it is a video game store. Or maybe you're like me and your favorite store is the grocery store. All right. Now, while you're picturing yourself in that big store, you're probably realizing there are thousands of things that they sell. I want you to picture a very specific item, your favorite one that they sell that you would want to pick up and take to the checkout counter. All right. Be very specific when you're doing this exercise because it's going to help us in the end. So, for example, if your favorite store is a clothing store and maybe you have a favorite sneaker that they sell, well, you want to think about the fact that it's a sneaker, its brand, its size, the color you want. Think very specifically. So I'm going to circle back to my example of the grocery store. And my favorite item that they sell is Haagen-Dazs chocolate chip cookie dough ice cream. All right, very specific. Now, let's think about this. I walk into the grocery store. This is a big store. They sell lots of different things, probably thousands of different items, okay? Lots of things fit into the grocery store, but I'm looking for that one specific item. Now, is the grocery store organized or are the items just kind of thrown everywhere and I have to search around for hours? No, there's a system in place to help us find certain things. So let's think about narrowing down the categories until I can get to that one item. So I'm in the grocery store. Where would be the first place I would want to go to? I'd probably want to go to the frozen aisle. Okay, so we've narrowed down my category. Now I'm in an area that has the Haagen-Dazs ice cream, but also has some other things. What are some other things you would find in that aisle? You're going to find things like frozen pizzas, frozen vegetables. You're going to find Eggo waffles. So although this category is smaller, there's lots of other things that fit in there as well. Let's continue on. Let's narrow it down a little bit further. Now maybe I'm going to go to the area that has frozen desserts. Okay, narrowing it down. All right, now is the Haagen-Dazs ice cream the only thing that's there? No, there's other things. Maybe there's ice cream sandwiches, popsicles, sorbet. So still other things are there. All right, we're going to narrow it down again. Smaller category. Let's focus in on the ice cream. All right, we've got all different brands of ice cream. So we're going to narrow it down once again to the Haagen-Dazs ice cream. And the last thing we're going to do is we're going to pick up that one item, the chocolate chip cookie dough Haagen-Dazs ice cream. All right, now this does relate to Carl Linnaeus' system because he had different categories that were very large and included lots of different organisms all the way down to a category that includes only one specific organism. So we're going to take a look at that right now. So I'm going to share my screen with you for a moment. All right. So these are the categories that we use when it comes to taxonomy, okay? So up at the top, you can see we have domain and kingdom, and those are very big categories that are going to include a lot of different organisms in them. And as we continue down the list, we have phylum, class, order, family, genus, species. I think it's always fun to come up with different sayings or ways to remember the order here. But as you get down to the bottom, you have the category species. And in species, usually only one organism is going to fit into that category. All right. So you've heard the word species before, and actually, you may not have heard of this, but you've definitely seen it before. There's something called a binomial nomenclature. A binomial nomenclature actually stands for a two-name system. If you've ever been reading in your textbook or maybe a book about animals and you've come across the name of an animal and then in parentheses there's a different name of that animal, it's usually in Latin, it's in italics, the font is kind of slanted. Well, that's the binomial nomenclature for that organism. The way they get that name is by using the genus and the species, which are those bottom two categories that you can see on your screen. All right, so let's take a look at an organism that we're probably pretty familiar with, Homo sapiens. That's the binomial nomenclature for humans. All right, so I'm going to just change my screen. Hold on one moment. All right, so here are the kingdom, phylum, class, order, family, genus, species of Homo sapiens. So you can see that the genus is Homo, species is sapien, hence why we get that name. But let's go through the other categories as well and understand why we belong to those categories. So our kingdom, remember, kingdoms are very big categories. And specifically, we're part of the animal kingdom. Well, we fit into that kingdom, but what else is going to fit into that category as well? Think about it. Whales fit into that category, beetles, snakes, fish, birds, lots of different things. It's almost like when we walked into the grocery store, we were in a very big category, okay? We're gonna narrow it down a little bit more. Phylum, chordata. Chordata actually means that we have a backbone. So we're starting to eliminate some of the other species that we were mentioning before, and now we're just focusing on species that have a backbone or a spine. All right, narrowing it down a little bit more. We get to class, mammalian, okay? So we're mammals. So that's going to only include species who are mammals. We're not going to include birds or fish in that category. And we're going to continue going down until we get to Homo sapiens, our species being sapiens. All right, now what we're going to do today is we are going to look under the microscope at two different specimens. We are going to be looking at cells that belong to the animal kingdom. Specifically, we are going to use my cells to look at. And then we're going to focus on a different kingdom as well. And we are going to look at plant cells. And we'll just see if we can see any sort of differences or similarities between the two. All right. So we are going to, before we prepare a slide, I'm just going to go to our next. And let's just quickly talk about some of the characteristics that would make something fall into the plant kingdom or something that would make it fall into the animal kingdom. All right, so first we're looking at plants here, and we can see some characteristics that they have. First of all, we see that they are multicellular, meaning that they are made up of more than one cell. They are eukaryotic. What does it mean to be eukaryotic? It means that our DNA is kept inside of a nucleus, or cells have a nucleus. Or I should say the plant cells have a nucleus. And then the last one is they get their nutrition through a process called photosynthesis. Photosynthesis is the fact that a plant can actually take sunlight and convert it into energy that the plant can then use. All right. And so those are some characteristics of anything that's going to fall in the kingdom of plants. But let's take a look at characteristics of things that will fall under the category of animals, including us. All right, so you may notice by looking at the slide that already we're seeing some similarities here. The first thing is that animals are considered multicellular. We are made up of more than one cell. That's the same as the plants as well. We are also eukaryotic. Our DNA is kept inside of a nucleus inside the cells. But here's a big difference is how we get our nutrition. Can I just go outside in the sunlight, lay out there and get the energy I need for for the day? No, I wish that would be great, but no, we get our nutrition through ingestion. Okay, we need to eat different types of food in order to get nutrition and energy for our body. All right, so I think we're ready to take a look at some different cells from plants and animals. All right, and first I'm probably going to focus on plant cells. Now, the way we are going to do that is I do have a microscope here that is hooked up to my computer, so I'll be able to share what I'm able to see. And I also have a prepared slide for you. So this one, if you can see it there, all right, has been already prepared. And this is onion bulb epidermis. What does that mean? So that's the skin of an onion. So that would be considered a plant. So we're going to take a look at these cells under the microscope, and let's see what we're able to find. So I'm just going to start us off by placing this on the stage. Okay, I'm going to turn on the light. All right, I always start on the smallest lens. That's the most zoomed out. I always think about if I were to have a paper with a whole bunch of words on it, would I want to start looking for one specific word with it back here or right up against my face? It's going to be easier to start further away and then bring it closer. All right, so I'm just going to first kind of put this in the middle. And now if you'll just be patient with me for a moment while I get the screen up. All right, awesome. We're already seeing something. Okay, we're just moving things around. I'm going to go the other way. And now we're just going to focus a little bit more, trying to get a little bit more of a clear view. All right, awesome. So what we are looking at here is plant cells. Now, these are pretty interesting. I always think that they look a bit like bricks that are laid down. They're right next to each other, and they do have a pretty strong structure. Now, why do they have this shape to them, do you think? What kind of organelle or extra structure do they have that animal cells would not have? They're going to have a cell wall. Let's take a closer look at these. Excuse me, I'm going to grab water in just a moment. Let's take a look. All right, that looks awesome. Okay, so I want you to think for a moment. There is a dot in the middle of each one of those cells. What do we think that dot is? Think back to what we just talked about. What was that? That's the nucleus of those cells. And what did we talk about that is inside of the nucleus? It's going to be the DNA. Awesome, very cool. This is a really good view of plant cells, nice and clear, and we're able to actually see an organelle within that. So that's a really cool thing. All right, now I'm going to stop sharing my screen for a moment. And we are going to prepare now our slide of animal cells. Now I told you I was going to use my own cells to do this. So what do you think I'm going to use? What type of cells am I going to use of mine? A lot of people think when I do this with my classes, they always think that I'm going to be using blood cells. That's not the case. All right, we are actually going to be looking at my cheek cells today. The reason why we use cheek cells is they're actually very easy to collect. They're very loose on the inside of my mouth. We just use a toothpick to kind of scrape them off. Doesn't involve any pain, no bleeding, nothing like that. So we're gonna take a look at that. All right, so the way we do this is I use a toothpick and I'm going to go into the inside of my cheek and just scrape in one direction downward. Then I'm going to take a slide, a blank slide, just like the one we were using, but there's nothing on it. And I'm going to take that toothpick and tap those cells onto that plate. All right, so I'm going to do it a little awkward, but it's in the name of science. And we're just going to scrape. Okay, and tap. And I'm going to tell you why it's important that we tap them down in just a moment. Okay, and tap. All right. Now, keep in mind that our cells are going to be translucent, so we need to add some sort of dye, and this is methylene blue. And I'm just going to dye my cells a bit and make certain structures stand out. So all I'm going to do, I'm just going to actually bring this down for a moment so you can see what I'm doing here. So I've got my prepared cell there, slide there. I'm going to open this up, and I'm going to carefully just collect a little bit of this. And I'm just going to put a drop right, ooh, there we go, a little bubble on there. Okay, I'm going to close this up because this does unfortunately stain, so we do have to be careful. All right, so now I've got my slide here, and what I'm going to put on top is a cover slip. All right, this is just a little bit of plastic that's going to go over. I just carefully, without trying to get too many air bubbles, I'm going to kind of just plop it onto there. And as you see, all that dye now spreads out a bit, which is a good thing. We want all of that to be spread out. And now my next step, I like to take my prepared slide and I'm just going to fold it up in a paper towel to get rid of any excess of the methylene blue out. So just carefully fold it a few times. You can see some of it coming through the paper towel. Press down a few times and then unravel it. Okay, so now my slide is ready to be viewed under the microscope. So let's take a look at it. Come back over to our microscope. I'm going to get rid of our plant cell slide. And now I'm going to put my slide that has my cheek cells on it. Okay, my lights. Okay, and now I'm going to share my screen again. I'm going to zoom out for a moment. Okay. All right. Okay. Now I do have a few air bubbles, unfortunately, in there, but that does happen. But what you're seeing here is all these tiny little dots are actually my cheek cells, which is really cool. I got a lot onto there. And we've got some pretty big areas with lots and lots of cells. All right, awesome. Just gonna find a good area where we can zoom in on and get a good look. Now, before I was telling you, make sure that you tap down your cheek cells onto the slide. The reason for that is if you kind of scrape them off, you can actually roll up your cells and they'll be like little burritos and you won't be able to see them that well. So that's why it's important to just kind of tap them down onto the slide. Let's take a closer look at this one. Give it a moment to adjust. I'm going to focus in a little bit more. All right, awesome. So these are really big clumps together. I'm going to see if I could move around just a bit and see if we could find some on their own. All right, awesome. So if you look at where I'm about to point my mouse, so around this area, this is a really great spot to take a look at. All right, each one of these is an individual cheek cell. Now, what are some things that you're noticing are different about these compared to the plant cells? Well, we're probably noticing that they're not stacked together like the onion bulb epidermis was. And they don't have the same shape either, okay? Remember, those plant cells are going to have a more structure, they're going to be more structured because they have a cell wall. Animal cells do not have a cell wall, all right? However, what are we noticing that is also in the center of all of my cheek cells? What is there? What is that dot? It's the same thing it was on our plant cell because we are also eukaryotic. So that is going to be the nucleus. So we are able to see where my DNA is kept within these cheek cells. All right, awesome. All right, I'm going to stop sharing. All right, so that kind of concludes up just our quick discussion on taxonomy, talking about how and why things are classified, and just taking a look at two of those kingdoms. We looked at the animal kingdom, we looked at the plant kingdom, and we were able to make some comparisons between the two. I hope you enjoyed. Thank you so much for joining us, and hopefully we'll see you again. Thanks so much. Bye.